Thanks, JB. You're making my life a little easier. Uh, so JB talked about the nature of variability and the sources of variability. I'm going to be presenting on behalf of Yibei, a postdoc in our group who couldn't be here, a way of assessing this variability. You need to be able to run all of these tools and pipelines. How do you do that efficiently? Okay, all right. JB showed this picture of the Rotem paper. There were multiple teams, multiple pipelines, multiple results. Analytical variability was a major contributor to the irreproducibility of some of the results in this paper. And there were many steps in pre-processing analysis that introduced subjectivity. And we do this all the time. We don't have perfect decisions in many parts of our workflow. So it's not enough to have a single pipeline. We need to quantify how different pipelines behave with different choices. But perhaps one thing that I won't be talking about today, because I'm going to be talking about a tool on how you could get to quantify these different analytic choices, is also qualify these different analytic choices. And I think this speaks to JB's point number 3 from that last slide, is when you have a problem at hand, what are the best choices you can make to solve that problem, as opposed to the easiest choices one can make to solve that problem? So to do this, we rely on standardization. To be able to run many pipelines, we can get to a state where perhaps our input data is standardized. Perhaps our containers or our pipelines themselves are standardized in a way that they can run on the input data, and it would give us output data that hopefully is also standardized to move the process forward. Because when we run various workflows and pipelines, the full complex nature from data to a result involves many different steps. And it's not just for those of us who do neuroimaging, not just pre-processing. There could be statistical learning, there could be statistical analysis, all kinds of things that happen along that pathway. One way to do this is we need some common ground on what we can do with input data. For those of you who haven't heard, is there anybody in this room who's not heard of BIDS? Please raise your hand. Fantastic. That tells us our community has come a long, long way, right? It tells us how data should be organized, and it helps us to reduce our cognitive load, to standardize, and make it easier for software to work on it. How many people have never heard of BIDS apps? Okay, there are a few. So just like BIDS organized things, BIDS apps were meant to work with BIDS data sets in a systematic way. This does not say that what's in the app. It was more about this interfacing, that if you have a BIDS data set, we can work with that data set in a systematic manner. But together, this allows us to kind of ask this analytical variability question. We can swap things around. We can change what's inside an app. We can change what configuration an app has. So I don't have to repeat that the organization of the brain imaging data structure helps us cognitively follow what's in our data. BIDS apps provides the same kind of conceptual framework for processing that kind of data structure. So the data sets are increasing. How many people here have worked with the UK Biobank data? Okay, I see a few hands. Is that your typical run-of-the-mill number of subjects you acquire? No. So as things scale, we need to change and make our processes a little bit more efficient. So there's more input data, but there's also commands and parameters. If we truly want to express this analytical variability, we want to change configurations on things. The apps themselves go through different versions over time. And one of the questions I ask people is, let's say a new version comes out, what do you do? Do you rerun all your processes? Generally not. It sometimes depends on what those changes are. But we are living in these complex pipelines where we don't understand what the nature of those changes could be. So somebody should be running these pipelines across versions to establish some of these differences. There have been some related efforts to help with this process. How many people here have never heard of DataLad? Let me get a setup. Okay, we've got a few hands. DataLad is a version control system that's built on top of Git and Git Annex that allows us to version data just like we version code on Git or GitHub. So you can maintain a specific version of your data set with DataLad. With Git and GitHub and Git Annex, we can also version code, so you can maintain specific versions of code that comes alongside it. And towards this effort, Michael Hanka and others in his group produced something called the Fairly Big Framework. But it turns out that this framework was kind of difficult to run for the arbitrary person out there. Most of us don't have access to the same kind of cluster or same kind of computational interface. We tend to use what's available, either in our lab, in our institution, and from that perspective, we wanted to figure out a way to make something that was easy enough to perhaps translate across different institutional variances. We also wanted to say, okay, we have single session data, multi-session data, raw BIDS data sets, so data that has just come from scanners, BIDS derivatives, because we also want to run things on derivative data sets. We have various kinds of pipelines that we might want to run on some of these things, and we might have different systems at our institutions that also either introduce variability in some ways. As JB mentioned, operating systems. How many people here have a cluster in their home institution that you use? Okay, that's more than 90% of this. How many people know which operating system runs on the nodes that you're running your things on? Okay, I see 4, 5, 6, 7. On the other side, about 10%. In many ways, some of these things, your partitions could vary. Your systems that are running can vary in their operating system version. You might know it's running CentOS or Ubuntu, but the versions could be different across some of these things. And with containerization, it's making it easier for us to run things very easily on any of these things. But as Jamie mentioned, the underlying bits that run the container could itself have impact on the outputs of things itself. So lots of combinations, not friendly to people getting started. To address this, we developed BABS, which was BIDS Apps Bootstrap Automation. The idea of taking BIDS dataset and being able to use different kinds of other BIDS apps on this efficiently. So I'm going to describe this a little bit today. So BIDS dataset, standardized input, identifies the subject, sample space, enables consistent comparison across pipelines. A config file specifies parameters and app arguments. These are the knobs you want to use to explore variability. And everything is saved in version control, so it should be repeatable, subject to randomness in processes themselves, which maybe Tom will talk about later on. BABS is a job generator, builds one job per subject, job runner submits to an HPC cluster, and it's an auditor. It tracks status, logs, completeness. All the things we don't really like doing, but we want it to be taken care of us so that we can understand later on what kinds of things could have affected our analysis outputs. And then the BIDS app output and an audit gives a directory per subject, log files, hashes, metadata, and can be compared across run for variability assessment. Overall, you do something like BABS init, BABS check setup, BABS submit, BABS status, BABS merge. There's a pretty well-configured set of steps you can run on your cluster. So let's take a use case. The Healthy Brain Network, it's a data set we play with all the time. There's about 2,500 subjects in this data set. And so you choose a data set. Maybe we want to run part of a workflow from fMRI prep, an anatomical processing workflow. And then you have parameters for fMRI prep itself. Now, while many of us do run fMRI prep in kind of parameter-free mode, we just run it with the default settings. It, like many other pipelines, can be configured for different kinds of approaches. And sometimes you want to configure them. So you can use parameters for fMRI prep. You run through Babs init, it creates a project, you check setup, it checks that you can run things, and then you submit jobs. And there's a tool called Babs status that helps you check how jobs are running. And I'll give an example of this in action in a little bit of time. I just want to go through these different steps in terms of how we prepare and submit these workflows. Babs status tells you what the state of the project is. So you run it on wherever you're running this setup, and the job status kind of tells you, oh, so many jobs were successfully finished, so many jobs are pending, so many jobs are running, and so many jobs have failed. Among all failed jobs, it tells you information about why they could have failed. And they could have failed for all kinds of reasons, right? A subject may not have had a T1 image in this particular case. Or if you're on a cluster, somebody kicked you out, kicked your job out when you were running this. Happens in clusters. So there could be many different ways in which you can have results, but you can also incorporate different kinds of checks on the system. So if you wanted to put in quality control checks, you could put in quality control checks into this system. Finally, after all of this is done, you call Babs merge, which merges all of these individual results back together. And this step creates your complete provenance tracked workflow result of the app that you have run. You can track everything from the original data down to the output through the container, and it's all checked in. And because this all uses data lad as an underlying system, you could rerun this process as if you started from scratch. Okay. Finally, it gives you back, in this particular case, a set of bits derivatives that you would also have available to be able to do further analysis or share with others. So I'm going to play a demo, and I'm going to voice over it a little bit. This repo exists. It's a toy demo so that anybody who wants to try this out at your home institution can go and start with this, and it'll walk you through every step, and you'll be able to do this in your system setup. So this is the repo of the demo. First, you clone the repo. This demo has been sped up to help with kind of going through this thing. You're checking out a branch. You activate a Babs environment. The actual environment setting is also in that repo. And then you start setting up some of these things. You have to prepare bids and data lad data sets for your containers. And what we're going to do now is create a container data set. So just like we're tracking code and data, we also want to track the container we use to run it. And what this will do is essentially capture the container we will use. And this is a toy container. There are any other container. And you'll hear later from Stefan on all the containers that are available in Neurodesk, so you could use any of those things. Then the next thing you do is you start configuring a YAML file to specify configurations related to your cluster. This is not something that anybody can do automatically because every cluster is somewhat different. You may have access to certain resources, and then once you've configured those things, you initialize this Babs project. This will run through, create a bunch of things for you, taking care of things behind the scenes. And once it's done, you will ask to check the setup. And if all was good, check setup will say everything was successful. And then you can submit your jobs with Babs submit. And you can see there were jobs, and you see that every job has failed. Happens, right? So now you can look at why the job failed, and you realize that there was something that was missing. And this system this repo walks you through some of it. It allows you to go and fix it. Now you submit it again, and this time around, you can check your jobs, whether they're running. You can check with Babs status, and voila, all your jobs have succeeded in this particular case. And now you call Babs merge. And what it will do is take all of that things that it ran and put it all together. And now You can see that it says Bab's merge was successful because everything had been processed and put together. And now you can check the different kinds of results. So in this particular case, because all of this is a datalad data set, you run a few datalad commands. I would encourage you to go through an extensive tutorial datalad has about how to use datalad. But for the moment, in this, we give you what you need to run. And once you run this, you can then see, hopefully it'll come up very soon, that all of your subjects have been processed through this very simple container. And you can fetch these subjects. And once you fetch the subject, because it's a zip file, you can unzip it. And now you can see that this container contains itself a bits data set, and it has the output from this container in there. And that's it, right? If you can figure out a way to run all of these steps, now you can start playing around with all of the pieces, whether you're changing data sets, whether you're changing pipeline parameters, whether you're changing the containers or pipelines themselves. Okay. I have left this slide there. I'm not going to talk about it. This, for those interested, this goes into some of the underlying technical details of how Babs actually makes the magic happen under the scene. And it uses something called a remote indexed archive, which is a technically tricky topic to make this work. But because of it, you were able to do and track all of these pieces and work in parallel across thousands of subjects when you're processing these things. Okay, so what might one want to do with Babs? You can obviously change app version. You can take something like fMRI prep with different versions and run it on the same data set. Remember our thoughts on standardization? Because these are both BIDS apps and you're running on BIDS organized data, you're able to connect the dots between those two things. You might choose different output templates or spaces. This is something we don't tend to do a whole lot of these days, but in the early days of imaging, we had created custom templates for many subject groups in various parts of the world. So you might want to look at what that might look like in the context of something like fMRI prep. You might also want to choose subsets of data based on quality control or other things. I mean, if we are going to process, UK Biobank is right now, I think, at about 65,000 subjects. We might want to limit them to a set of subjects that don't have any quality issues. So that might itself be a quick check on the system when you're running these pipelines. And then you might want to vary output metrics. You might want to look at motion, FD, co-registration quality, TSNR. And these are some of the variants you could run. But most imaging pipelines are way more complex and have way more parameters than what I'm showing up over here. And I think as a community, we have to understand where these parametric choices happen and what their impact is, not just on the output, but why we choose them for our specific use case and question. Okay, that was a repeat. Oh, no, I'm going backwards. That's what. Okay. So finally, you might get a hypothetical comparison workflow, fMRI prep through config A, fMRI prep through config B. You might be able to check different things. Hopefully in your use case, it's not that the brain has been flipped around anterior, posterior between the two use cases, but that could happen. There could be a bad data set out there, right? So to summarize, the challenges were, how do you run so many workflows with so many variations? How do you track all of these changes exactly as you're running these things? And you'll hear from some of the other platforms today on how they are doing some of these things. We wanted something that we could run in our own institutional clusters quite easily, so abstracting out some of the concepts and places. So now you get version-controlled execution of BIDS apps. You get a structured audit trail of all commands, inputs, and outputs. It's scalable across data sets, sessions, and configurations, and makes the data finally ready for analytical comparisons. It tracks all of the provenance details that you now need to see what had actually happened. So Babs has been a partnership between Ted Satterthwaite's group at Penn and the ReproNim project. It's out there, you can use it. We have this demo repo, so you can practice how to use this on a very toy problem just to get it set up, and then you can adjust and switch to your data sets. With that, I thank you for your attention and can take a few questions. Yes. I can hear you, but I don't. Yeah.
I'm wondering about, so let's say you go through the process, you bootstrap a few pipelines. You also mentioned that you also have tools there for analysis. So I was wanting to hear a little bit how would you analyze, let's say you have many pipelines, and of course you have many features and many results. What is the way to compare those? What's your thinking?
So I'm going to start by saying that I think it depends on the question you're trying to answer with the pipeline. So I'll make up a question to answer your questions. Let's say you're looking at some relation between some brain activation state and some behavioral metric. Maybe I want to look at, I don't know, network prediction of depression severity. I'll take that as a question, right? So when I do that and I want to see, did fMRI prep A versus B have an impact on it, I run through the whole process, right? All the way through to predicting depression severity. And now the question is, how do I compare these two things? Tom and I were just having a conversation about a comparison of two machine learning approaches at the end of the day. And this is like two models run on the same data, giving rise to slightly different results. How do you say one is better than the other? So when I look at this, I might use Tom's method that he's been developing to do that comparison. Maybe he'll tell you about it. Maybe it'll come out soon. On the other hand, I also want to know, how do I validate the outputs themselves, right? Because an analytical variability, I said, was, yes, you can change these knobs. But you also want to ask, why am I changing these knobs? And so I think it was Christian who gave this example with the ADHD 200 competition almost a decade back at this point in time, maybe more, where somebody who got the results, one of the highest results, was primarily getting it because of motion in the subject. So in this competition, they were predicting who has ADHD and who does not from brain imaging data. And it boiled down to motion as the source, as a primary feature of interest to that. So we want to kind of look at our pipelines through the lens of scientific validity as well. So when we look at it, we can compare and ask, are our results valid at different stages due to the knobs that we were processing? And because this is being kind of captured, you can go back and look at different stages of your pipeline. For any complex pipeline, maybe you're touching things in different places. If you're touching knobs early on in the process, that's going to have many downstream impacts. If you're touching it later in the process, that's going to make it easier for you to compare some of the things. So it really kind of depends on which pipelines and the question you're having. For a machine learning problem like prediction, I'm always interested not just in how well something did, but can I explain why that turned out to be the case? Hope that answers some of where you're going. Okay. All right, let me go back.
Thank you so much. Related to the question, in that case, since Babs can be used for doing this sensitivity analysis, I think, on how your pipeline affects your results and stuff, how do you suggest maintaining transparency and reducing the investigator bias in terms of reporting results in publications?
Great question. I think that's exactly what Babs is suited for, because all of those methodological changes are tracked in that repo at the end of the day. So if you publish that repo, which doesn't mean publishing the data that comes along with it, but it can publish all of your configurations, all the changes, all the evaluations, kind of exactly. And if somebody else, so let's say you did do this on a public data set, then somebody else could take that repo and rerun that exact same thing. So A, it addresses reproducibility, and B, because it addresses reproducibility, it also addresses transparency. So I would encourage, if you use Babs, to kind of think about publishing that final data set that you have curated through the process out there. Now, there are some challenges I will highlight when you do that. You have to make sure there is no PII data, personally identifiable information, anywhere in your configuration space, right? So that's something that I think we need to be aware of, whatever analysis we are doing. So as long as that's true, then you can send that data set out there, and people would have access to that full transparency. Now, there could still be biases, right? My choice of parameters is my biased view of how I see the world in terms of analytics. Hopefully somebody else can look at that and go, no, Satra did not choose the right parameters here. I'll tweak it and create the next few pipelines that addresses a better space for the question he was trying to answer.
Thank you so much.
Okay.
So can they use data that is not downloaded by data lab, like their local data? Is it going through the data container that you mentioned earlier?
So what we often do is, if we create a bits data set, it's very easy to make that a data lab data set. So the starting point, we have reduced everything to, you need a data lab organized data set, and you need this data lab organized container. Once you have those two things, then the rest of it can make assumptions about that space. But it's very easy to create a local data lab data set. Okay, all right.